Okay, hello everyone. I'm, I'm sorry for the noise. It's noisy on the train station right now. Please ignore. My name is Kyoko Aoki from Stanford University, and I will be moderating today's APBioNETTalks. Just to give an introduction, APBioNETTalks is an online education program. It's been initiated by the Asia Pacific Bioinformatics Network, or APBioNet, and it's been happening since the middle of October 2020. This program aims to facilitate open access to knowledge sharing of bioinformatics. We provide both live and on-demand research presentations, tutorials, hands-on practicals, and demos across bioinformatics fields. Experts, early career researchers, scientists, and students from different countries and institutions are invited to share about their research, projects, and skills with the audience. As for now, we collaborate with the InBioSys, UKM Malaysia, and BioGlues to provide content for our audience from Asia Pacific, Europe, and Africa. The program encourages contributions from communities and institutional partners. Both pre-recorded and live videos are published on the APBioNet YouTube channel. As you can see, this one is being streamed live right now, and you can find more information on the APBioNet homepage. Okay, and with that, I would like to introduce today's speaker, Daniel Bojar. He received his PhD from ETH Zurich, and after working there as a researcher, he was a postdoctoral scholar at the Weiss Institute at Harvard University. Currently, he is the Associate Senior Lecturer for Bioinformatics at the Department for Chemistry and Molecular Biology and the Wallenberg Center for Molecular and Translational Medicine of the University of Gothenburg in Sweden. He also is a recipient of a Branko Weiss Fellowship at Society of Science, and he was also selected as one of the Forbes 30 Under 30 Europe list. So he's a very promising young researcher, and I'm very excited to invite him to speak. So Daniel, the floor is yours.
Thank you very much for this nice introduction. It's a great pleasure to be invited to speak to you today. So I try to make my talk as educational as possible in the sense that I am not entirely sure about the backgrounds of all of the listeners. So I have quite a lengthy introduction into my subject of choice, glycobiology, so that we are all on the same page. If you have a background of any manner in glycobiology, you can sit back for the first couple of slides and relax. So with that, let me already get started. So our focus is very much at the interface of computational research and glycobiology, seeing what we can do with methods that are specifically geared to investigate glycans and their role in biology. But in order for you to appreciate that, you first need to really get a detailed sense of what glycans and glycobiology are. So I want to start at the very basics. So first off, what are glycans now? Because that is a frequent point of confusion. When you mention glycans to researchers, they frequently have a dim understanding, if at all, of what these molecules actually are. And you can see here a very illustrative example from just 2 years ago by now, where the CNN, the magazine, had just titled glycans as a type of protein, which obviously they are not, but that shows you just a characteristic misunderstanding at the level of knowledge that you can commonly expect to see in even the life science community. And that's why I think these slides are necessary, because glycans are important on the one hand, but underappreciated and unknown on the other. So I think that's a potent, but also a situation that requires introduction. So just at the very, very basic chemical level, glycans are biopolymers or biomolecules in the broader sense. They're essentially composed of sugars. So they are long chain sugars. That's where, or it depends, it can be medium to long chain sugars, depending on the molecule you're looking at. That's why it's polymers. Biopolymers is because they are being produced in biological systems, actually in basically all biological systems we know about. So everything from viruses to bacteria to archaea to animals to plants, they all carry glycans on their surfaces, on their proteins, etc. On the left here, you see the actual one chemical constituent of a glycan, a so-called monosaccharide. This is more akin to the simple sugar that you put on your cake or whatever, and this is sort of the building block of these longer molecules. And you encounter them all around you, actually. So the more maybe commonly known glycans or carbohydrates, as they can also be called, are cellulose, chitin, starch. So these are by mass. They make up not only most of the glycans, but also most of the organic molecules per se. And you can easily appreciate that by just going outside and looking around you, and you can see that most of what you see is actually carbohydrate material. So that already gives you a sense of the pure structural role of glycans and just literally upholding everything we see around us. That is also true on the molecular level. Glycans also have an important structural role, a shaping role, a modulating role on the protein level, on the cell level, on the organism level. As I mentioned, they're composed here of simpler sugars that you arrange in different combinations. So they can really be compared to the other biological sequences that we see in biology. So if we group DNA and RNA under nucleic acids, then glycans would be the third type of biological sequence. So DNA and RNA, obviously very well known, they are composed of 4 building blocks, and there's only one way that you could connect 2 building blocks. So that gives you a sequence that can be very, very long, but quite information poor, because the options are just so limited. You can have very repetitive regions, etc. That's different for proteins, where you have 20 building blocks already, the amino acids, these canonical ones, and you can have up to 2 ways to connect them if you consider cis and transes, 2 different geometric means of connecting amino acids. And that gives you a lot higher diversity. Consequently, the sequences are shorter, because you have already a lot of diversity, and you don't need to go to these extreme sequence lengths to convey your information. Glycans are more diverse still. They have more than 100 building blocks with an asterisk that I will go into in a second. But more importantly, these building blocks can be connected in numerous ways. So the exact geometry, how you connect 2 monosaccharides in a row, is very different, with different biological consequences. And this increase in diversity means that you need even shorter sequences of glycans to convey an information-rich message and have biological consequences. The asterisk that I mentioned is that these more than 100 building blocks do not occur in the same cell. So one of the many peculiarities of glycans is that they have a non-uniform alphabet. So in more or less every organism, you will encounter the same nucleic acid building blocks, the same protein building blocks, but you will not encounter the same glycan building blocks. So every organism, to some extent, has their own unique repertoire of monosaccharides that are only partially shared between species. So that is also a difference to consider. And just one example to show you that these tiny differences really have huge consequences, biochemically speaking. I wanted to pick out a disaccharide. So this is a molecule that consists of 2 glucose molecules that you may be familiar, because that's actually one of the sugars that you may encounter in normal life. These 2 glucose molecules are connected via a so-called glycosidic bond. This is just how you connect the monosaccharides in a chain. And the difference between these 2 molecules is the orientation of the hydroxyl group of the first glucose. So it's alpha in the first case, it's beta in the second case, as you can see here in this text representation. But that makes all the difference. So the left molecule, maltose, it's the building block of amylose, which is a component of starch. It forms helices, and we have, as humans, have enzymes to degrade maltose. The right molecule is cellobiose, so the building block of cellulose in plants. Cellulose does not form helices, it's a linear molecule. And we do not have enzymes as humans to degrade cellulose. So that already shows you that this tiny structural difference has a lot of consequences for biochemistry and for eventually function and properties of the resulting molecule. And you can play that game ad infinitum almost. So here are some of the glucose disaccharides all have different linkage patterns, and they all occur in different circumstances, have different properties, etc. You may have, for instance, encountered trehalose before, which is used as an anti-freezing agent, and so on. And that's only a disaccharide, just as a reminder. So I need to show you some nomenclature so that you don't get lost in later slides. So you will see a lot of these pictorial representations. This this color shape combinations. This is the so-called symbol nomenclature for glycans, or SNFG, and it's a defined pattern of associating each monosaccharide with a unique shape color combination. So the glucose that I've talked about in the last slides would be a blue circle, whereas a green circle is a different monosaccharide, mannose in this case. So it's not super important for you to know each combination at this moment. It's just for you to know that each shape here represents a monosaccharide, and the lines in between are the linkages. So how they, which monosaccharide is connected to which. If you are sharp-eyed, you will notice another peculiarity of glycans as a biological sequence, and that is that they form branch structures. So you see here, this is a non-linear arrangement of building blocks. This is unique in comparison to DNA, RNA, or proteins, which are strictly linear. So another factor that increases glycan diversity, structurally speaking, and makes it harder to work with them, obviously. The last important point here on this slide is that glycans have two ends. So the one end is called the reducing end. This is the part where glycans are connected to something else. So for instance, proteins are, as you will see, are heavily glycosylated, so they're modified with glycans, and the modification occurs at this end of the glycan. The other ends are sort of the business end of the glycan. They are the non-reducing ends, which are exposed to the surface, to the solution, and can interact with proteins or cells or whatever. Just for the sake of completeness, there are lots of other depictions of glycans as well, not super relevant right now, just for you to know that there are also text-based descriptions here, which you may encounter in later slides. Some are human readable, some are designed for computers to process these sequences. But just shows to you that it's an ongoing challenge to depict these branched, diverse molecules in a useful manner. Another important point to consider for glycans is how they are made. And that really is again a point of uniqueness for these type of molecules, because DNA, RNA, and proteins are part of this central dogma of molecular biology, where DNA is transcribed into RNA and RNA is translated into proteins. It's a code-dependent, template-dependent process. I don't want to say that glycans are completely non-templated, but it's largely non-templated in the sense that there is no strict code that tells the cell to make glycan A on protein B. Rather, it's a conglomeration of enzymes that add or remove one monosaccharide at a time to these glycan trees, and thereby shape the final molecule. In animals, it's usually happening in the secretory pathway, so the endoplasmic reticulum and the Golgi apparatus. That means also that most of the secreted material of a cell is being modified with glycans. So meaning glycoproteins, for instance, you may have heard, but also lipids, etc. And what for me is really crucial here is that the fact that glycans are being produced by enzymes in the secretory pathway really means that it's a very dynamic process, because you can change the expression or the activity of an enzyme and thereby change the resulting glycans. So you can very, very quickly change this type of molecule without having to go through genetic mutations, etc. But on the other hand, you have the fact that this is a biological sequence. So you have a lot of diversity, you have a lot of information density. So you are sort of at this point where you have the best of both worlds of being dynamic, but also being information rich and diverse. Because if you compare this to something like phosphorylation, which is a simple on-off switch, you would have the dynamic nature, but you would not have the information density. And for humans, this is really quite drastic. So the estimations differ, but it's definitely around at least 2/3 of the proteins that produced and secreted by the cell that are being covalently modified by glycan sequences. And those sequences in turn shape protein folding, protein stability, protein function, etc. So there's a lot of modulation there going on. And that doesn't stop with proteins. You also have lipids, you have glycosaminoglycans, you have a lot of sugar cover around the cell. So this is a bacterial cell, for instance, which is very popular in slides of this topic to show you this fuzzy hair almost around the cell, which is the glycan forest, if you wish, around the cell. And this is really important because if you think about a protein can be glycosylated at different sites. So you can have different amino acid positions in the protein that can be modified with glycan sequences. And importantly, each of these sites typically has different glycans. So that is what is being referred to as macroheterogeneity, whereas in microheterogeneity, it's referred to the same site with different glycans. So you have two levels of heterogeneity, which result in basically a distribution, is what you get out. So if you think about one popular example is prostate-specific antigen, so PSA, which is a cancer biomarker. If it goes up, it's one explanation could be that you have prostate cancer. Another explanation, though, could be that you have prostatitis, so prostate inflammation, which also causes PSA to go up. So it's a biomarker, but it's imperfect. What is better, however, is to use a so-called glycoform of PSA. So every protein that is secreted, or most proteins that are being secreted, are a distribution of different glycoforms. So where a protein has a specific glycan sequence on its surface, changing its properties, as I mentioned. This distribution changes in disease conditions. So not only does the total expression of PSA go up, but also the glycoforms that you can observe behave differently. Some new ones may appear, some old ones may be upregulated, etc. And these are usually more precise biomarkers than the total protein expression, because these are the ones that are actually functionally relevant. So when I talk about this in the extended format, I always like to quiz people here. I won't call you out, but I want you to take a second to think about these three entities and think about which ones of them do you think are glycans. And I'll give you a quick second to think of your answer this for yourself in your head. The reason why I do that is because as you've seen, hopefully, with the cellulose and the chitin and the starches, that glycans are all around us, but we don't realize. And that's, I think, the big tragedy of the field, because it is relevant for everyday life. So if you take the first, blood groups are maybe the most salient example of glycans that actually constitute something that you maybe talk about quite frequently in your life, because the blood groups themselves are literally glycan sequences. So which of these exact glycan sequences you exhibit on your erythrocyte determines which blood group you have, at least with the classical APBio system. And that is in turn dependent on the expression of certain enzymes that produce these sequences, of course. Biofilms are, so I don't want to say, so it's not that all biofilms are glycans, but some to many biofilms are predominantly composed of glycans, and the water that they bind, forming this sort of hydrogel-like structure. So very important for antimicrobial resistance in that sense. The last one is sort of the false friend of this quiz. Glycogenin is actually not a glycan but a protein, though the saving grace here is that glycogenin is involved in starch metabolism, so it has some relation to glycans, therefore the name, but it's not a glycan itself. Okay, so coming closer to the actual the actual research part here is sort of the broad question of what does it do, right? I mean, so okay, glycans are there, they are very diverse, but what do they actually do on a functional level, on a mechanistic level, what is the biological importance. And the example I took here first is a very well characterized example. This is the effect of glycan sequences on antibody function or potency. So antibodies, or in this case here immunoglobulin G, IgG, are of course a major form of defense of our adaptive immune system. And they are being glycosylated because they are a secreted protein. So meaning that you have a distribution of glycan sequences on these IgG molecules, and which sequence exactly you have determines the properties of the antibody. So you can see here one very well characterized example, for instance, is if you lack this red triangle over here, then the antibody has characteristically pro-inflammatory activity. It is usually just a very strongly enhanced activity. The red triangle in this case is the monosaccharide glucose. The specific motif would be called core fucosylation, and the lack of this really increases antibody activity, which is why it's also used in biotechnology by dedicated research for pushing the antibody into a state where you don't have this core fucosylation motif. Another example would be heparin. Heparin is a common anticoagulant, so a blood thinner, and is a glycan. It's a glycosaminoglycan, so a long chain carbohydrate actually. And as you can see here, it's heavily sulfated. So so a lot of sulfate modifications. But the important part is that where and how many of these sulfate groups you have in the molecule dramatically changes its properties. So how potent is it, and what kind of pharmacokinetic properties does it have? So that's it's almost like a sulfate code. And for a long time, this heparin was extracted from pigs. So as you can imagine, any natural product that you extract has a lot of diversity, has a lot of batch-to-batch variability, especially if it's so finely tuned with these sequence-to-function relationships of the sulfation patterns. So that causes problems. Nowadays, it's synthesized in a controlled manner, so that's no longer bad. But it's important to keep in mind that these tiny differences can have large effects. And that's really sort of at the core of it, that the sequence of these molecules really matters a lot. And that's usually because you have the recognition system. So in most cases for glycans, that's proteins. So you have proteins whose dedicated function it is to bind specific glycan motifs, specific glycan epitopes, which can be used in self recognition or also be leveraged by microbes, by pathogens, in a process known as molecular mimicry to pretend to the immune system that it's actually a self structure, but and therefore evade the immune system attention. So the sequence matters, but the problem is how to get from the sequence to the function. And the difficulty really here is that not only are glycans super diverse, but they're also hard to work with, both experimentally as well as computationally. For all of these and probably more reasons, which is why it's so challenging for most researchers to incorporate glycans into their research. And that's sort of where we want try to come in to from the computational angle, to provide tools, methods, and approaches to somehow make sense of these molecules, both mechanistically in detail, but also at scale. Because you can imagine, if already single sequences give you trouble in determining their function, you will have major problems if you look at it from a systems scale. So this is sort of like our overview slide of what we do. So we continuously built a lot of methods to analyze and process glycans and to somehow work with them. These are combined in our open access Python package GlycoWork, which is available on PIB and GitHub and wherever. So if you are somewhat familiar with Python, You should have an easy time to get into this. GlycoWork not only contains basically all of our methods, but also trained models that I will talk about later, and also a lot of data sets that we curated from various sources, and that GlycoWork uses as an internal knowledge base to power a lot of the functions. Okay, so the scientific core part here of this talk will be around these proteins that I mentioned that bind glycans specifically. And the reason for that is because not only are they important in our bodies, but they're also important as reagents even. So most labs, or many labs, at least depending on what they do, use glycan binding proteins, or lectins as they're also called, as reagents to stain tissues, to stain cells, or any kinds of other things. Because if they bind something specifically and a certain cell type expresses this epitope, then you can, of course, use this as a staining reagent. You can also use it as a therapeutic, because if a glycan epitope is overexpressed in cancer, then you can use a lectin for that epitope to direct a therapeutic against that cancer. In our body, you will predominantly find lectins in the innate immune system. So here are some of the proteins in the innate immune system whose main or sole function is to bind specific glycan epitopes on fungi, bacteria, viruses, etc., and deal with them. But of course, you have that also on the other side of the divide. You have microbes, you have viruses who have proteins, again, whose dedicated function is to bind particular glycan epitopes. And in the case of microbes and viruses, that's of course for the purpose of adhering to cells, entering cells, infecting hosts in the end. That gives them their species tropism, that gives them their maybe tissue tropism in some cases, and is in general the first point of contact. So for viruses, for instance, even if they make contact with proteins, they first make contact with a specific carbohydrate receptor as a first step. So you might, if that didn't convince you already, why it would be interesting to study lectins. It's also a very much ongoing effort. So here is a PNAS paper from last year, which actually determined the carbohydrate receptor for an important immune checkpoint receptor, Siglec-7. So that's a human receptor, and until last year we did not know what it actually was binding. So it's still an ongoing process. As I mentioned, these molecules, these proteins are being used very, very frequently in staining assays. So it's of course important to know what they actually bind to if you want to make conclusions based on these staining results. And you can even use lectins themselves as a method. So here this is called a lectin array, where you basically glue lectins onto a slide, and then you add samples, and you can thereby profile which glycan epitopes are present in your sample via fluorescence, etc. And lastly, as I mentioned, you can use these molecules directly as therapeutics or diagnostics, or any kind of other translational application where glycans play a role. But the main question that ties up our projects that I'm presenting here is really, can you tell from the protein what it will bind? Because that's sort of ultimately the goal, right? I mean, in nature, in the lab, you would encounter a protein and you want to know what it binds. And of course, there are lots of experimental methods of going about that question, but that they may or may not be scalable. They may or may not be feasible depending on what you're looking at. So we wanted to go about this from a computational angle. And we started doing that with a collaboration with Lara Mahal from the University of Alberta in Canada, and looking at 57 of the most commonly used lectins. They're just listed here, a bunch of abbreviations, not important. But we really wanted to get into the nitty-gritty and see, can we do a detailed annotation of their binding specificities using a combination of interpretable machine learning and expert annotation. And the idea is really that we use data that is generated from glycan arrays. So in contrast to lectin arrays are sort of flipped. So you glue glycans to the slide, and then you add the lectin. And the lectin, in our case, is biotinylated, so you can add streptavidin, fluorescent streptavidin, which then visualizes where the lectin has bound. So in this case, this schematic lectin will be specific for sialic acid, this purple diamond, which you can then figure out via analysis. And we use different vendor options of these molecules, different concentrations, etc., just to have a broad coverage. And the process really is then, so for the interpretive of machine learning, is that you try to learn rules. So the way how you do that is because if you a priori assume that the lectins bind epitopes, so fragments, motifs, pieces of the glycan sequences, then these rules should be looking at the presence or count of these motifs in the respective glycan sequences to find out whether the lectin will bind. So the model itself learns many sub-models which apply logical rules. So we'll go through an example which makes it hopefully clear. But basically, is motif A present or absent, or present in more than 2 copies, together with many other rules that are being applied in conjunction, and with that, then predicting whether a lectin will bind that glycan or not, based on the glycan array data. So if you take the example of MEL2, which is a tree lectin, as you can see here, it's one of the list of 57. The set of logical rules that are, so the top performing rules basically that help in predicting whether it binds or not are these two. So this sialylated motif has to occur more than, or at least once, so more than 0.5 times, and this it's called core 1 motif has to occur more at least once. And those are 2 independent rules, so those come directly out of the machine learning model. But then when we combine this with the expert annotation, the presence of core of this motif indicates that it's a certain class of glycan, namely O-linked glycans, the type of protein glycans. So the expert synthesis is then that this lectin is specific for this, specifically alpha-2,3 linked sialic acid on this particular class of glycans. And indeed, in this case, MEL2 does not bind this glycan motif on other types of glycan classes. So it does seem to be specific for this subset, which is important if you want to use it and want to properly interpret the results from your assay. And we did that for all 57. I won't go through all of them, of course, just so we try to group them in a manner that is useful for users, so that you can actually go and have a look if you want to detect a certain epitope. You can see what is the best choice for that, at least from the commonly used ones. So just 2 examples to illustrate you why this is important and what you can learn from that. The first is MEL1. It's a very similar name, different lectin, same tree. No. MEL1 is also used as a specific probe for alpha-2,3 linked sialic acid. So it's commonly used similar to MEL2, basically. And here are just a few randomly chosen papers which confirm that. So where authors specifically state they use it as a specific probe for this epitope. The issue, however, is if you use our rule-based approach together with the expert annotation, you get this motif as the top binding summation, which is a sulfated structure, and is obviously already visually different from the motif that is being used as a reagent form. And of course, it does bind sialylated sialic acid, no question about that. It's just that this one binds better. So if you do get a signal with MEL1 as a reagent, you cannot conclude that you have this motif present in your sample, because it could be that it rather binds to that one or a similar like that. Just important to be aware of that. The second example is wedge A. It's a lectin from wheat. It's commonly used as a specific probe for beta-linked GLICNAC. However, it is being recognized that it also binds other things, predominantly salicylic acid, but it's still frequently used as a at least semi-specific probe for that. If you look at the rules for determined binding towards in our approach, you see that it's actually quite a bit. So not only beta-linked lignac and salicylic acid, but also alpha-linked lignac and galnac and N-acetylmoraminic acid and lots of things. So you might think this is the antithesis of a specific binder. I would argue that it's the opposite. It's too specific. And it's because it's specific for a particular chemical moiety, this N-acetyl group that all of these 5 monosaccharides carry, but none of the other commonly encountered salicylic acids do. So that's why it's seemingly unspecific from the spectral representation, but very specific if you look at it on a chemical level. Again, just important how you use it, how you interpret the data. That's why we made the study in the first place. So it's great that it's possible to do it in this much depth. But the issue is really that it's quite a convoluted process. You need a model for each lectin. You need the human in the loop. So it's not very scalable. You need data for each specific lectin. So we need this lectin array data for each lectin. And in nature, there are not 57 lectins. There are hundreds of thousands of lectins. So that's a potential issue if you want to be thorough. So we did a separate approach in collaboration with Frederick Lisacek from the Swiss Institute of Bioinformatics, and to make a deep learning based approach to have a generalizable model that can predict lectin binding specificity just based on protein sequence, which we termed lectin oracle. So the idea is that you give lectin oracle a protein sequence, you give it a glycan sequence, and you get a binding prediction. And since it's really easy to just get protein sequences, it's the idea is that it's very scalable, very generalizable, and hopefully performant. So to do that, we first need to make a sort of a detour of how do you actually do deep learning on glycans. And if glycans would be like that, it would actually be not a huge issue because deep learning on biological sequences is by now quite established. There are lots of deep learning models on DNA, RNA, proteins, etc. The issue, however, is that most of these deep learning methods treat these molecules as text, as language. They use natural language processing-based models predominantly to analyze the sequences. But glycans, as I mentioned, are not linear. They cannot be treated as left to right text. They are branch sequences. They are non-linear. So if you do the mental model, they are basically like trees. And trees are just a special case of graphs. They are graphs that never close in on each other, on it again. And that's really great because natural language processing is a huge field, of course, but graph theory is also a huge field. So there's a lot of already established machine learning, AI, deep learning approaches that work specifically on graphs. So learning graph neighborhoods, learning graph properties, and using them for prediction. So, and one area where that also has been established is in the small molecule area. So in drug development and predicting drug properties, small molecule functions, etc., where you basically learn features from the molecule as a graph. So, for instance, this neighborhood here would be characteristic in endowing the molecule with certain properties. You also have, of course, subunits or chemical motifs almost. So in the chemical case, that would be maybe you have a chloride group here, or you have a hydroxyl group somewhere. All that changes molecule properties, all that changes the graph structure. So you can learn that. The same is, of course, true for glycans, in the sense that the specific graph structure of a glycan is indicative of its properties. May not be a straight linear mapping, but at least it's something that can be learned. So this is why we applied graph neural networks to the analysis of glycans, so that we can extract information from the glycan as a graph and then use that to predict properties. So in the case of lectin oracle, we use that for the prediction of the property of glycan binding. So that means we need information from both the protein and the glycan. We use again as a training source the glycan array data, but instead of using dozens of experiments like we did in the previous approach, in this approach we use thousands of experiments. So that's more than 500,000 measured protein-glycan interactions, which hopefully is enough to generalize to new molecules. And then the model is taking in separately basically the protein information and the glycan information. The protein information, we don't need to reinvent the wheel, so we just use a pre-trained language model that uses the actual protein sequence and extracts then the features that can be used for binding prediction. Whereas for the glycan information, we use this graph neural network that I mentioned before. And then extracted features from both moieties are combined and then used to predict the actual binding. So you get, in the end, you get a number that you either can threshold to say binding, no binding, or you treat it as a semi-quantitative measure of how well protein A will bind glycan B. And if you just take this with an arbitrary example here, this is the lectin SNA. This is one of maybe model lectins, if you wish. It's a super well characterized lectin that is actually super specific for alpha-2,6 linked salic acid. So on the x-axis, you give lectin oracle all kinds of different glycans that predict the binding to SNA, and then which is depicted on the y-axis, and you can see that the glycans that are predicted to bind to SNA actually are highly enriched in this in the motif that is already known to bind to SNA. So that's great, but that's of course a cherry-picked example. I don't expect you to be satisfied with that. And more importantly, we wanted to see whether we can actually generalize to new proteins, glycans, experimental environments, etc. So we went about validating it. The first step that we did is go back one step and actually look at the 57 lectins that we characterized together with Lara and see would we have gotten the predominant binding rule with lectin oracle. And that's especially important because 19 of these lectins are actually not contained in our training data set. So that's good because that shows us whether we can generalize to new sequences. And indeed, for over 90% of them, we would have detected binding to the predominant binding motif. So that's already quite encouraging. But we also gathered more data sets from the literature where people have designed their own glycan arrays, so using other proteins, using other glycans, using other experimental setups. And we wanted to ask how quantitative are our predictions, with the caveat that glycan arrays themselves are not perfectly quantitative. So it's always semi-quantitative in the end. But by basically correlating our predictions with the actual measured binding. And here are just 4 examples from 4 different experimental assays. And it's not a perfect correlation. We wouldn't expect that because it's semi-quantitative at either end. But we do routinely get moderate to strong correlations of the measured binding and the predicted binding, which is good, especially because this is just a naive linear correlation. And as you can see, for some of them, it's decidedly not linear. But that's very encouraging because it shows us that we can indeed apply this model to new sequences on both sides of the divide, potentially generalizable. We then wanted to see one of the typical things that you do with a well-performing model. What did the model learn, in a sense, right? So did it learn structural binding features of the actual lectin-glycan interaction, or where does it get information from? So for that, we used, here as an example, the protein sub-B. It's a lectin from E. coli, which binds salic acid. And indeed, the native version is predicted by lectin oracle to bind salic acid. If you now go ahead and in silico start to chip away from the known binding site, which has been characterized by structural biology, you very quickly lose the predicted binding to salic acid. So it's not perfectly point mutation aware, So there's still some work to be done, I think, because experimentally you already lose binding with the point mutations here. So we do not see that here. We see it if we take away the slightly surrounding area. So again, not a perfect correspondence, but still quite encouraging that there seems to be some recognition of the actual binding site in the prediction. You can take that one step further because one thing that people like to do with lectins, or that is quite promising, is something called lectin engineering. So changing the protein, changing the lectin so that it has a new binding specificity via directed evolution, probably at some point via protein design, but in the end, introducing mutations into the lectin to change the binding site, etc. So in this example, they took here a natural lectin, which binds galactose glucose disaccharide, and changing it so that it also accommodates here the sialic acid moiety. So it's now a new lectin, or at least a lectin with a new binding specificity. So we wanted to see, can we contribute to that with lectin oracle? Can we use some kind of in silico screening? Can we accelerate, etc.? So we wanted that. For that, it's of course important to be aware of the impact of mutations. So we first needed to see whether we are. So a different lectin engineering experiment. Here they wanted to engineer it to bind a sulfated structure, where the wild type binding is here, and then each mutant has the associated binding affinity or relative binding to the sulfated structure. Some mutants increase binding, some mutants don't show an altered binding. So those are just some examples that they chose in this paper to show how the directed evolution proceeds. If you take these sequences, which are very helpfully made available in the paper, so that's great. You can take these sequences into a lectin oracle because we are generalizable, so we can just take any sequence into the model and get predictions, and you plot what the predictions look like. It's quite encouraging because you see that basically, except for one mutation or one set of mutations, all the mutations that increase binding to the desired motifs also are predicted to increase binding to the desired motifs, while those that do not show a change in the binding also do not show really a change in the binding in the prediction. So in this case, at least, there's a very good match of the engineering approach and the predictions, which could mean that we can in the future maybe use this to boost this method further. One very specific example is we wanted to see, not only can we make new lectins, but can we find new lectins? And the reason for that is that there is this great database called Lectome Explorer, made by Frederic and Anne, which is a database of predicted lectins. So these are hundreds of thousands of lectins that have been found in genomes of uncharacterized species and all kinds of organisms. No one has characterized them, of course, because it would be insane to do that for hundreds of thousands of lectins. So we wanted to see, can we use lectin oracle to screen this database for lectins with potentially interesting binding behavior and then characterize them, which is a lot better than characterizing them at random. The one we chose in the end is a lectin from sweet melon, Cucumis melo agglutinin. That's why we christened it CMA. And the first thing that you do is, of course, we order the gene, we produce the protein, and then we send it off to like an array experts in Boston and in the Imperial College in London. And they then tested the binding of CMA to, in the end, over 1,000 ligands. And that's really, really great because this CMA really shows quite a unique binding specificity that we are in the process of further investigating, namely C2 substituted galactose, which is a bit of an abstract binding specificity, but very interesting. We are currently in structural and biophysical characterization of this protein. Okay, and then there's a last vignette, sort of as a going more on the system scale, is we have the possibility to analyze lectins at scale. So we wanted to actually show that we can use that here in the case of the microbiome, where we, again, due to the collaboration with Frederic, we could use her workflow to use, in this case, the vaginal microbiome. She then extracts via hidden mark model the predicted lectin sequences that are being produced by the vaginal microbiome, and they collaborated with experts in the vaginal microbiome to classify the strains into are those commensal strains or pathobionic strains. So what you can do now is you can take these lectins, we put them into an lectin oracle, we predict their binding, and the first thing that you can do is compare pathobionic and commensal strains. So what you see already is that pathobionics, the lectins of pathobionic strains, not only do they have more, by the way, but also the ones that they do have show a broader binding specificity. And that's exactly what you want if you want to adhere to tissue. Just because it gives you more things to catch onto. So that's definitely something that could contribute to their pathobionic qualities. We also can have a look at what they actually bind. And what we do find is that in the pathobionic strains, there is a shift towards more human-like glycan epitopes, particularly fucosylated structures, a lot of so-called Lewis antigens, which are quite characteristic and important. That again should contribute to a better catching on to the mucosal surface for the pathobionic strains, and potentially also give you then a modality to block that binding by adding something that exhibits these glycan motifs. And then as the last vignette is viral infection here. Viruses, as I mentioned, bind glycans to enter the cells. So in the case of influenza, that's hemagglutinin, the protein hemagglutinin, which binds salic acid. Avian influenza binds alpha-2-3 linked salic acid, very broadly speaking. Mammalian influenza binds alpha-2-6 salic acid. And that is really dependent on the sequence of hemagglutinin, because if you, let's say, give avian influenza a mammalian hemagglutinin, it would suddenly bind alpha-2-6 linked to salic acid. So it's again sort of our core assumption that the protein sequence of hemagglutinin determines its binding specificity, which we can use. And that is not only different between avian and mammalian hemagglutinin, but also within each of these classes. So the changes in the sequence changes the binding properties. We use that in Taiwan, or with data from Taiwan, I should say. So where they very helpfully made available all of the strain information from 1981 to 2007, which we used then. We used in the hemagglutinin sequences to predict the binding to human-like glycans. You see some fluctuations over the years, depending on which strain was dominant. Very well matching the experimental binding of these strains. And then if you overlay the excess mortality that was due to the strains of that influenza class, that you see at least some indicative match with the increased binding. So again, the hypothesis is that if it binds better to human-like glycans, it could be that it's better at infecting or at least increasing the viral load that is being imposed on these patients. So this definitely needs more investigation, but it's an encouraging first result, I think. And with that, I'm at the end. I want to, of course, like to thank my group and our collaborators. I always give a shout out to Black Work. Feel free to try it out and let me know. And thank you for your attention. I'm happy to take any questions.
Thank you, Daniel, for a very interesting presentation with lots of information, including the very basic glycan information. It's great. Thank you. So are there any questions from the audience? I had a question about when you look at the WDA. Can you hear me okay?
Yeah.
Okay. So it looked like from the monosaccharide level that the binding was very different, but when you look at the atomic level, you can see the same similar pattern across the polysaccharides, right?
Mm-hmm.
So would it be possible to create a more lower level learning model at the atomic level as opposed to the monosaccharide level?
Yeah, definitely, and I think that that's really needed in the end. And so we have some preliminary results on that. And interestingly, if you just use an atomic level, in our hands at least, it underperforms the token level models. But if you combine both information, it outperforms the token only model. So both levels are needed, and if you combine them, you get indeed higher performance. So that's definitely the way to go, I think.
When you say you combine them, does that mean that you use both in the same as a feature vector, or how did you combine them?
So it's again this two-arm approach. So where each arm looks at the glycan from a different resolution, basically, and then you combine the feature vectors that are learned from each level.
Got it. Also about the lectin.
I think that there might be something in the chat. I don't know.
Was there?
Or maybe not. Oh no, I don't think there are.
Yeah. Yeah. Any basic questions, even if they're very welcome. Anyone in the audience, any of the students? There's a question. Have you observed any specific order of binding of these glycans, especially in the virus?
We haven't looked at order of binding actually, so that would be interesting. Especially because I mean most of these lectins have multiple binding sites, which in some cases even have different binding specificity. So those would be the most prone to exhibit an order of binding, and I don't know of an example where this has actually been shown to show like a really specific order of binding or multiple binding sites. So unfortunately, I don't have any insights to share, except for that it's a really good point.
Also, I know that looking at glycan arrays, that there's also questions about non-binders, and you can predict the non-binders as well in your model.
So for the in-depth approach in the collaboration with Lara, we had that in a sense with Lectin Oracle. I mean, we implicitly get those, we just don't do anything with them. So that's a good point. So it would be important for people to know, and especially because I think what is especially tricky for people to appreciate is that if the same epitope can be a binder and non-binder depending on the context of the sequence, right? So it's in a way, enrichment is really important to know that maybe the one instance of the epitope that you see highly binding is the only instance of this epitope that binds highly, and usually it's not. It's a non-binder. So that's really important to appreciate, of course.
Yeah, the context would be important. Question from Hilya: what are your thoughts on deep learning application in terms of data size and facilitated automated learning from the data?
That's really good, especially because it will become more and more important. Because so if you take glycans only and try to predict properties from glycans, data size is actually not an issue at all, because we don't have the billions of sequences that the people in protein science have. It's more like tens of thousands, or up to 100,000 or whatever that we have, and they are of decent size, which is so that is definitely fine. But if you now go into the direction of something like an Oracle, where you include protein sequences, suddenly that increases the data size and the model size and everything by a lot. So basically pulling in more and more other data types can also be transcriptomics or whatever, which is, I think, very much needed, and can give you great advantages, is something that will also increase the need for computational infrastructure. But again, it's nothing at the moment, at least. It's nothing that that you couldn't even do with almost free resources that are currently available. So you don't need to be DeepMind to enter the field at the moment, at least.
Okay, and then there's a question about online tools or databases for prediction of glycan structure and effects on gene or protein targets.
So specifically the first point, there definitely are tools for prediction of glycan structure. I mean, GlyCam is definitely one option which is available. There's a lot of molecular dynamics simulations which has done great work both on the actual glycan structure, but also on the effect of the protein that the glycan is on. So especially in the context of the SARS-CoV-2 pandemic, there is a lot of really good research on the impact of the glycan shield and the impact on the interaction of SARS-CoV-2 with its receptor protein.
There's also the glycoinformatics consortium, which you are leading.
Great point.
Which would might have more information about informaticians in the glycomas field.
So I see a
have
question about
C-type lectins and targets.
Yeah, we have worked actually quite a lot because the innate immune system is full of them, basically. So of course there are special interests both for our group but also for a lot of the community. And the really cool thing is of course that there are quite a few C-type lectins that have glycan array data. So meaning that our model should capture their binding quite well, because so that's a general thing about machine learning models that you have to keep in mind, right? So that you can only expect to get good performance if you have seen something similar, at least to some extent, in your training data. So therefore, I would argue that lectin Oracle should be able to do well with C-type lectins. And I see a question about graph modeling. So definitely the performance would definitely change depending on which graph model you use for the protein glycan interaction. We currently use a graph convolutional neural network, which at the time of making lectin Oracle was the best in terms of performance. There are several other graph neural networks that show at least similar-ish performance and similar both in terms of actual accuracy as well as how long it takes. I think by now it changes what the goal is basically. So for instance, when we do atomic models, we observe that the models that use attention work better than models that use just straight graph convolutions, because you have a lot. The graphs are a lot larger, right, on the atomic level than on the token level, because suddenly every atom is a node in the graph. So that's different requirements. But in general, you should get quite decent performance with most graph models.
Great, thank you very much. I think we're about time. So I think we'll close the discussion now. Thank you very much, Daniel.
Thank you. And thanks, everyone.
Ilya, could you? Yeah, thank you. So we just wanted to make some final announcements. Ilya, would you like to do that?
Yeah, sure. Can we perhaps take some photo together?
Group photo, yes. So if possible, if people could show the video, we would like to take a photo.
Right. So maybe on my mark. One. Share your biggest smile, and if you don't mind, turn on the camera. I think I'll turn on myself. All right. Thank you, everyone. 1, 2, 3. Yeah, for documentation. Another one, another group. 1, 2, 3. Okay, thank you so much, everyone. So back to you, Kyoko. Shall we announce about the next APBioNETTalks?
Yes, could you do that? My internet is kind of weak.
Okay. Okay, so let me just share a short video.
Yeah.
Yeah, okay. Let me share just a short video for the next talk.
Maybe you can show your screen, the video.
All right, just a sec. So the next APBioNETTalk will be on 17 October. And then we also have INCOP on 21 to 23 November. Still have some days left to submit your paper to INCOP. And then another one is from INSIP. This is more for students. And you still can also submit the paper for INSIP. It will be on November 5 and 6. That's it. That's from me, Kyoko. Back to you.
Okay. Yeah. No, I'd just like to thank everyone for attending, and I hope to see you again at the next APBioNet talks. Thank you very much.
Thank you. Goodbye.
Thank you, Daniel.
Thank you, everyone. Bye-bye.